and established a working area of novel drug delivery systems by handling various formulation development in Natco Research Center. He supervised 36 MPharm students, 9 research projects in PhD, and supervising one more project. He worked on various DBT, UGC, CSIR, AICT sponsored research projects. He also worked on biotransformation of ether lipids at Institute of Lipid Research Monster, FRG, under Indo-German Academic Exchange Program. He worked as Vice President at Natco Center and as a Chairman, Board of Studies in Pharmacy at Kakatiya University. Sir received Best Paper Award from Kakatiya University in the year 2004 and 2005, also at IPC Mumbai in 2006. He received fellowship from German Academic Exchange Service to work in German laboratory during 1987 and 88. He published around 36 papers in reputed journals and presented 27 research papers in various national and international conferences. He visited various countries like Germany, UK, Switzerland, Canada and USA for research and business-oriented programs. Ladies and gentlemen, let's welcome Dr. Shashank Sridhar Apte sir with a big round of applause.
So, good afternoon. Before I begin, a little bit about me. As I was introduced, I was working in Kakatiya University. I worked almost for 25 years there. And after that, I joined Natco Pharma. I was fortunate all throughout. In Kakatiya University, I could see the best of the period where I learned a lot and all the basics. I could gain a lot of understanding during my period in Kakatiya University, the period which I spent in Kakatiya University. In Natco also, when we joined, we started a new area of research wherein Natco was interested, novel drug delivery systems, particularly nanotechnology based products. And we started from scratch. We were shown an area, an empty area, and I said, you do whatever you like. So we started from there, and not only the development of, the basic development, conceptual development of the product, but design of the plant, selection of equipment, the entire design of the line, manufacturing, training the people who are actually undertaking manufacturing, regulatory part, then to a certain extent marketing, that means advising our marketing people, and also giving presentations to doctors. So the entire thing I was fortunate to get exposure to. Initially, I remember when we developed the product, scale up was required, and for that, some equipment, particularly some vessels, etc., were necessary. So, I started interacting with the people. I had never seen a vessel which was used in industry, particularly in injectables. And I started interacting with the fabricators. I just told them what I wanted. But then there were several issues, like what kind of wall you need. I said, please tell me what are the different kinds of walls available so that I can select one. I mean, that was the situation, and I feel I'm very, very, very fortunate to have undergone all through these things. So I felt that whatever the experience I gained, probably put in a form so that And all through, I believe it was the observation, very keen observation and the analysis that helped me to be successful in industry as well as in academia. So I chose this topic because this is something which is undervalued. Nobody pays probably this is one quality which cannot be quantified. I mean, you cannot assess the power of observation in a person. But then, what I understood and what I experienced is observation is the key for not only the problem solving, but also for innovation. People nowadays talk a lot about innovation, and maybe based on my experience, I will tell you how the power of observation contributes to innovation. Can you take it forward, please? Is to observe, listen, and capture the details. We must understand that observation is not just a casual look. Observation is more than that, and it is about paying attention to the details, patterns, nuances that often go unnoticed. I'll just give an example. You are handed over a floral bouquet, and if I say that observe it for 5 minutes, and then I'll ask you questions. You observe it carefully for 5 minutes. What are the different kinds of flowers? What are the colors? Different colors, how many colors are there and all. And then give back the bouquet to me. The question I ask is, what are the different types of leaves that you find in there? Nobody pays attention to the leaves. Everybody pays attention only to flowers. So, the fine things often go missing. And it is these fine things that lead to solutions. These fine things that lead to innovation. So, I'll give some examples of problem solving and some examples about innovation and how observation helps in these things. So, a simple thing, we go, you get a document, and you just go through that. You are asked to evaluate it, you are asked to review it. You just go through that and you say everything is fine. This is an example of a procedure wherein you go through that and you say that everything is fine, I can make this. I can follow this procedure. But then, when you really start following that, you will have several questions which you never had earlier. What could be these questions? Yeah. Next. Yeah. Sample. Now, how much amount of sample? The first question you get is how much of sample you have taken. Then next, aqueous phase. Again, the quantity of aqueous phase. How much? Next, in a jacketed container at 60 degrees centigrade under stirring. Everything is explained, but a jacketed container at 60 degrees centigrade means whether the water that is circulating through the jacket was at 60 degrees or the product you want to be at 60 degrees. So, jacket temperature is always different from the product temperature. Next, The mixture was extracted. Now before that, the stirring. What kind of stirring was there? Then the mixture was extracted. Next. Using 50 ml chloroform and 100 ml methanol. Now, it was heated and then subsequently followed by extraction. Whether extraction was done at room temperature or it was done in hot condition, that is not clear. Next, 50 ml chloroform and 100 ml methanol, when you mix with certain amount of aqueous phase, you get a single phase. How are you going to separate the phases? How are you going to extract in a separating funnel? That means something is wrong. So something is missing there. Probably the amount of water which was intended to be added later was missing. And when you add water, you get two separate phases, and then you can go ahead with the extraction. So a simple thing, but many things are missing in this. You cannot really follow the procedure unless you know all these things. Next. Yeah. We may get a question as to why we should observe. The research says that observant people are better able to learn new information and understand complex problems and make better decisions. This is what the research has said. Another thing that has come out of research is observant people are happier and less likely to bear negative thoughts. And the most important is, it's a crucial tool in problem solving, fostering innovation, and it opens our minds to new perspectives and alternate solutions. I'll explain this with examples. Next, please. So while observing, one has to be neutral, absolutely neutral. One should not be biased, one should not have personal feelings or judgment, because this will lead to bias. Second thing is, observation should be from a distance. Many times you are involved in an experiment, but when you observe, you distance yourself. You believe as if you have not done it. You are observing it from a distance, and that gives a different perspective altogether, detached from the preconceived notion and biases, and it enables us to see the new things in a new light, from a new angle altogether. Next. Yeah. So I'll give one example which we have experienced. So I'll just give a background of that. We have experienced several problems we experience every day. So I'll give just a glimpse of that and what was the problem and how the problem was resolved. So the procedure is, powder is dispersed in water, so it's a lipid powder. It is dispersed in water, heated, and passed through a membrane under pressure for size reduction. So this is what we do for making liposomes. So I've worked on liposomes. We've made several products on liposomes. So this is the procedure we follow, right? Powder is dispersed in water and heated, and it is passed through membrane under pressure. So the pressure is given by the pump. We use a pump in between. Now what was the problem? The procedure is wonderful. What was the problem? Next. We found black particles settling on a membrane. So this was unusual. There is nothing black anywhere, but still you see very fine black particles settling on a membrane. So then the investigation started. Next. What are the possibilities? Where from these black particles could have come up? The possibilities are powder and or water is contaminated. There's no possibility of water getting contaminated because water is WFI, and we examined everything, the entire line. Absolutely no problem with water. powder is contaminated, that is a possibility. Contamination from the lime could have been contaminated with something, maybe some rust somewhere. Some moving part is getting eroded, that is a big possibility, and some external contamination. Now if you go back, please go back. Again, yeah. If you look at the line, you have a vessel, which is stainless steel vessel. There's a stirrer over there. Then a pump and a membrane holder. So what are the moving parts in this? The pump and a stirrer. So first of all, we started examining the pump, and we found that there is nothing moving except for a diaphragm, which is made up of PTFE. So other than diaphragm, there is no moving part. And then we contacted even the manufacturer of that pump. He said there is no such possibility of generating any particle in the pump. So pump was excluded. Then the other possibility is stirrer. If you look at this is a type of magnetic stirrer. I can show you the details of that. Go to the next slide, please. Next. Yeah. So this is the magnetic stirrer. So there's a rotor, which actually next one, please. So there is a shaft which is coming into the vessel. So this is the bottom of the vessel. So the shaft comes into the vessel. It is sealed, and then the rotor gets fixed on that, and it runs through magnetic power. And what you can see, that black thing there, that on the shaft, that is a bush, and the rotor moves onto the bush, but it is lubricated with the product. Suppose you have aqueous solution or aqueous dispersion, so that aqueous dispersion forms a lubricating solution for that. So there is absolutely no possibility of any particle generation unless you have abrasion happening there. So we examined from every angle. We examined, everybody was more or less certain that powder is contaminated. I asked our engineering people to have a look at the mixer, magnetic mixer. They opened the vessel, had a look at the magnetic mixer, particularly this particular portion, and they gave green signal. They said there is absolutely nothing wrong with this. So then the powder is the culprit. But then what is wrong with the powder? The powder also did not have any particle in that. So ultimately we came to a situation wherein the root cause is not found out. What could be the root cause? Everything is fine, but still the particles are there. So then I was at a remote place. This was happening in the plant and I was at home. So I was asking over phone as to, let us examine this once again. Although the engineering people have given green signal, nothing has happened to this. They were almost about to bet on this. But I just asked them to open and see. Then they saw had a careful look at it once again, and I said there is absolutely no problem. So I just asked one of them whether it is smooth, that particular bush, whether it is smooth or smooth and shiny. They said it is absolutely smooth, there is no issue. I said, you didn't understand my question, whether it is smooth or smooth and shiny. They observed carefully and they said it is smooth and shiny. Now the shin comes through very fine movement of the rotor, which rotates over that, and that was the reason for generation of particles. And the particles were very fine. They were in fact nanoparticles. Our product was a nano product. The particles were also nanoparticles. But still, they were settling onto the membrane because the membrane pore diameter was very small. So, ultimately, I asked them to do an experiment because nobody was willing to believe that this is the culprit, even if it was shiny. So, I said, let us do an experiment. Let us not stir it. Let us just mix it and then pass it through the membrane. They mixed and passed through the membrane and found everything absolutely fine. There was no particulate contamination. So ultimately, the person said, yes, I understand now, I'll replace this. So, very fine observation leads to solution. Smooth and shiny. The difference between these two, how you interpret the variations, such small observations, actually is the key for problem solving. Next, please. This is typically what is done in industry for problem solving. To get in the investigation for finding out the root cause. It is the Ishikawa diagram which is often used. Men, machine, material, method, mother nature, and ultimately that leads to the effect. So everything is investigated. Men, whether the people who are engaged, whether they are properly qualified or not, whether they have been trained or not sufficiently. Then machine, whether the machine parts are fine, whether the machine has been qualified, whether preventive maintenance has been done, and all these things. Material, anything wrong with the material, the raw material. The method of analysis, for example, or the procedure, how you take up the manufacturing of the product, and the mother nature, that is environment. So all these things are taken into consideration, but many a times what happens is, this is a tool. So it gives the result provided what you feed is proper. If you miss, for example, in this case, machine was the problem. But if the man misses what is wrong with the machine, then you don't have any solution. And what you do then? You just have to say that nothing was found, and this entire tool becomes useless. So many a times, what happens is, a very useful thing becomes useless only because you are unable to operate it, or you are unable to use it properly. And observation helps here. Next. Yeah. Again, observation involves engaging all our senses and being present in the moment. We can give valuable insights and spot opportunities and identify risks. Let me give an example. Another thing is, what you see, you have a thought behind that. If I see something, I will have a thought behind it. But that thought is not constant. Many a times, what happens is, you can change the thought. You can change the way of looking at the thing. You can look from a different angle altogether and you find a different thing. The scene looks the same, but the way of you looking at it changes, and this leads to solution. Design of experiments, or DOE, is another tool, and we had a training session on DOE, a wonderful training session. The trainers from Mumbai were training a group of our people, and they give, just to explain the things properly, they give some some examples, and they were just trying to establish that there is a relation or no relation through DOE. DOE is all about establishing a relation between the different parameters that we use with the effect. So, whether one parameter is responsible for getting the effect, or whether two parameters together are responsible for getting the effect, and so on. So, that is what DOE reveals. But again, this is a tool. So, they gave a wonderful, a very interesting example. Two persons going for a walk, and while going, they see a girl coming, and the girl smiles. So two persons, A and B, going together, and they find the girl smiling at them. Then everybody started thinking, each one of them started thinking that the girl smiled at me, A as well as B. So they thought of doing an experiment. The next day morning, B went along with C. This time the girl did not smile. So B felt that probably I am not the person, it is probably A. So he sent A along with C. Again, there was no smile. So A is also not responsible. He said, I am also not the person, and probably it is because of C. Because C is accompanying both B and A, one at a time. Probably his face is ugly, and that is why the girl is not smiling. So that was the conclusion. So again they went together, A and B. Again the girl smiled. Then they thought, let us resolve it once for all. And they said, okay, let me go alone. So A went alone next day. The girl did not smile. B went alone the very next day. Again the girl did not smile. So that means, ultimately, they wanted to convey through this example that A and B together are responsible for the effect. It is not A alone or B alone, but A and B together are responsible for this effect. But many a times, when we design the experiments for DOE, many times we select the parameters in such a way, probably some unimportant parameters we select, and ultimately we get some relations which are inconclusive. So during that session I asked him the question, and I gave an example also. Next one, please. That suppose the situation is like this. A and B go together, they can extract smile. B and C go together, they can extract smile. A and C go together, again smile. A and B together, again smile. A smile, B smile. So that means there is a relation of everything with the effect. There could be another thing, that the girl doesn't smile at all. So there is no relation. Now in such a situation, there is another angle to this. And it is possible in this case that, and it is very much possible that the girl was mentally retarded and she smiles at everyone. So this angle one forgets, and therefore they find relation with everything, but actually the relation doesn't exist. So you have to be very, very careful, and this is a tool again. Design of experiments is a tool. So you have to select your feed. You have to select what parameters you are going to select, and what effect you are trying to relate with that. If you do it properly, then you get some meaningful conclusion. Otherwise, you don't. Many times, many of my colleagues from industry told me that their experiences that this is useless. It doesn't give anything. Probably this is the reason. That they have not understood properly, and that's why they could not establish any relation. Next, please. Embrace curiosity. Approach every situation with a sense of curiosity and willingness to learn. As Dr. Krishna said, he learns from students. In fact, I like him, I learn from everything. Even now I am learning. Every day I am learning. So learning is a continuous process. That cannot stop. But the willingness to learn is important. Ask questions, challenge assumptions, and seek to understand different viewpoints. If somebody is saying something, you should give patient hearing to that and try to understand what he is trying to say, whether you can extract anything from that. Otherwise, if you don't listen to a person, then you won't understand his viewpoint at all. So, just to give an example as to how a curious person can make a difference. Now, everybody probably knows, at least the B.Pharm, late B.Pharm and M.Pharm students, they know about the homogenizer, stator-rotor homogenizer, right? So, it is something like this. There's a drive unit and then there's a probe. Now, this probe gets inserted into a beaker containing, let us say, dispersion. Suppose you want to make an emulsion, you have oil and water together in the beaker, and then you just insert the probe inside, give some RPM, and stir it for a certain period of time, or homogenize it for a certain period of time, and then you get a fine emulsion or a coarse emulsion, depending upon the RPM and many things. Now, as far as this part is concerned, it is fine in the lab. But if you want to scale it up, what is that you have to do? Is it scalable? You may say that I will have a bigger homogenizer probe and a big beaker, and then it will do the same thing. When you have a bigger probe, first of all, one must understand that it is not the RPM that is responsible for homogenization. It is the tip speed that has a role to play. So the tip speed is much bigger because of the diameter in large homogenizers compared to small homogenizer. So the RPM has to be higher in smaller homogenizers, very much less in larger homogenizers. Then the gap between the beaker wall and the homogenizer shaft, the bottom also, that is also important to give reproducibility and scalability. So this method of homogenization is not scalable at all. So if you think properly, then you understand that these things are not scalable. Now what is the solution for this? Next. There is another type of homogenizer which is called as an inline homogenizer, wherein you have a stator rotor inside a chamber, and then you feed the product from one side and get the product from the other side. This is scalable because the stator rotor remains the same. Only thing is that you feed the product and take it out. You can change the number of cycles to get the product, fine product or coarse product, whatever the way you like. So you can simply change the RPM, fix the RPM, and change the number of cycles. So this process is scalable. The earlier one is not. So this can be imagined. Scalability can be imagined. This is just one example. Next. Yeah, all in all, I would say that the problems are very difficult. When you face a problem, it is absolutely very, very difficult. It looks to be difficult. But then, next, the solutions are simple. When you find a solution, only this, when we developed our first product, it took us some 6 years to develop the product. We developed the entire process. It's quite a complex process. When we went to the plant with the process, and when we demonstrated that this is the process, everybody was saying, it took 6 years for you to develop this process. It is such a simple process. Huh? And 6 years. I said yes, because every step in that, you have to think a lot. And it's a kind of innovation at every stage. So, solutions are often seen. Next. Now, as I said earlier, observation is a crucial tool in problem solving and fostering innovation. Very, very important quality for innovation. It opens our minds to new perspectives and alternate solutions. Now, just to demonstrate this, I'll give another example. These are all live examples. Nothing is imaginative. So we conducted an experiment. The point was, actually, we had a class 2 drug, BCS class 2 drug, and we wanted to increase its dissolution. Rather, we wanted to increase its solubility, and thereby the dissolution and therefore absorption. So that was the objective. So we wanted to make nanoparticles of that, and that's why we tried first homogenizing the drug itself using a high pressure homogenizer. We could not get anything. We could not get fine particles at all. So we used another way of doing the same thing. So our procedure was, the drug is precipitated from a solution in a water miscible solvent by adding in the aqueous phase, so a non-solvent. The aqueous phase is non-solvent in this. The drug is in solution form in some water miscible solvent. Aqueous phase is a non-solvent here, and in the aqueous phase we added a surfactant and or a polymer. So we added a surfactant or a polymer or both. So the moment you add the drug into this aqueous phase, you get a precipitate. Now this precipitate is homogenized to see the extent of size reduction. This was the experiment. So the objective was size reduction. So we wanted to make some fragile precipitate such that it gets size reduced considerably when homogenized. Simple homogenization did not work initially. So we wanted to do this. And during the procedure, my colleague who was working on this, he made several observations. One of the observation was, with the drug, when a particular surfactant was used, he was getting a sticky precipitate. And the sticky precipitate it's not possible to homogenize that, because it sticks everywhere, it sticks to all surfaces. So it was sticking to the homogenizer and nothing was coming out, only water was coming out. So he said, no, this is useless. I examined that and found it very interesting. Why you are getting a sticky precipitate? Can the sticky precipitate be suspended? So we used a polymer along with that, and we tried to actually disperse that sticky precipitate into a polymer solution. So when we used a particular polymer, it was possible to disperse it, and we got a fluffy precipitate in that. So we examined that precipitate under microscope. Next. So this is the first photograph is that of the drug itself. The second and third, when we observed them under the precipitate under microscope, we started getting some unusual things. We started getting some fibrous structures, thread-like structures. So we were thoroughly interested as to what these thread-like structures are. And moreover, when these thread-like structures were subjected to homogenization, we started getting very fine particle size of 60 nanometers. Very fine particle size. So we were very much excited to find out as to what this precipitate contains, what are the different components in that, and how it looks like, this thread-like structure. Next, please. So the first picture is that of the drug microscopy, scanning electron microscopy. The second one, low magnification thread-like structure. And when the magnification increased, you find those thread-like structures which are porous. Every thread is hollow. And it looked very fragile. And when it was homogenized, the last picture, we got nanoparticles. And the size of those nanoparticles was somewhere around, average size was around 60 nanometers. So then we thought, yes, we have achieved what we wanted. So let us go ahead. Let us do some studies. And we studied saturation solubility next. The saturation solubility of the drug, as it is, was found to be 1.44 microgram per ml. And the nano formulation, we made nano formulations out of that. So nano formulation 1 and 2, we got 29.9 and 26.46 micrograms per ml. So almost 20 times increase in the saturation solubility. So this typically happens when you reduce the particle size. This is well known. Then we looked at the dissolution profile. In 10 minutes, the first formulation gave 90% of the drug, almost more than 90%, 93% of the drug getting dissolved. The nano formulation 2, it was relatively less. And the physical mixture of that was only 3%. So 90% versus around 3.5%. But then there was another thing that is happening. You can see that there is precipitation. So initially the drug dissolves, it forms a supersaturated solution, and slowly it starts precipitating over a period of time. Next, please. Then we did some animal experimentation, some biostudies there in animals. We fed to the animal the nano formulation 1, nano formulation 2, then a marketed formulation of the same preparation which was available in the market, and just to look at the fed-fasted variability. In the fed state also, we fed the formulation 1, and you find a huge difference in the AUC. The green one at the bottom is the marketed formulation, and you find that nano formulation 1 and nano formulation 2, they are much way above the marketed formulation. And in the fed state also, it is more or less the same. So fed fasted variability could be nullified and the bioavailability could be increased. So all this, had we not observed or had we not analyzed why that stickiness is there, I would have, anybody would have just thrown it out. A sticky precipitate, just throw it out, ignore it. So if you ignore, if you don't observe properly, we would have missed all this. So that's the power of observation. So in the innovation also, observation is important, and the analysis, observation and analysis together. Next, how thinking of that helps in problem solving. So this is from the literature, a solid oral product they wanted to prepare, and already in the market, a branded formulation was available, so they wanted to copy it, a generic formulation they wanted to make, a company, and they started making a formulation which would be bioequivalent. But the problem was they could make a tablet, but the tablet was not bioequivalent. Next. So when the dissolution was carried out at pH 1.2 and pH 6.8, you find that at pH 1.2, they are more or less equal. In fact, the marketed formulation was showing lesser dissolution compared to the developed formulation. At pH 6.8, also, they were giving very little dissolution, but then they were together at pH 6.8. So, looking at this, generally it happens in industry that on the basis of dissolution, you design you design a product, compare the dissolution with the innovator, and then go for biostudy. But biostudy reveals something different. When they did biostudy, they found that they were not equivalent. So they cannot claim that it is a generic product, they need to redevelop it. So this kind of thing is very common in Indian industry. I would say that I have seen in some industries, not in Natco, but I have seen in some industries, they made something like 15 to 20 attempts to resolve these issues. Every time, reformulate that, to do something, some combination, some excipient, increase, decrease, or some component, make some changes, and then reformulate and give for bio, because the dissolution always passes. So, your changes and you will get dissolution. You will be, yeah. So when the investigation was done, and they concentrated on physicochemical factors. So one of the factors they felt is important was the effect of excipients on the particle size of precipitated drug in the GI tract. So obviously, what happens in the GI tract is important for absorption. The nature of particles, the particles are generated which undergo dissolution. So the nature of particles formed probably is important for establishing a relation between biology. So normally for poorly soluble drugs, when a tablet or a capsule is made, and if it undergoes disintegration, you get typically large solid particles which are from 5 to 100 microns, and because of lower dissolution rate, the absorption is affected. Whereas, if you make a solid dispersion, and this was a solid dispersion, if you make a solid dispersion, then upon disintegration, you get fine particles. Next, please. There is a possibility that these particles, some of them at least, could be in the nano range. And next. So this was the question, that can it yield the nanoparticles? So this was the question which they asked themselves, and then they started finding out what is the range of particle size which they get. So using a typical master sizer, they did this experiment, and they found that the innovator profile is quite different from their own profile. The innovator profile, that is ACT30, that had particles in the range, if you look down the scale, 0.1 to 1 micron, there was a significant peak. 1 to 10 micron also a significant peak. And then majority of them between, somewhere around 50 to 1000 microns. So this was the profile of particle size distribution they got in the innovator. And in their case, what they found is the first bump was missing. That means the particles which were small in size, they were missing altogether. Even in the second bump, that was also to a certain extent less. So probably they thought probably this is the reason. And then they did formulation changes in such a way, they increased one particular polymer in that from 1% to 10%. And then after reformulating the product, they did this profiling once again, dissolution profile, particle size distribution during dissolution. And next slide please. And they found that when, see this is granule 1 and granules 10, that means 10% of the excipient and 1% of the excipient. They were using 1% of the excipient, then they started using 10% of the same excipient. When they started using 10% of the excipient, they found it was mimicking almost the profile that was obtained using the innovator product. So, next. So, once again, they made a comparison. After 10 minutes of mixing and after 30 minutes of mixing, the profile was more or less the same. Then they went ahead with the bioavailability, bioequivalence studies, and they found, next please, almost superimposing curves. So it was bioequivalent. So just by looking at the things from a different angle altogether, a different perspective if you use, then probably it leads to solution. If you look at the things plain from on the surface of it, and every time you keep looking at the things on the surface, then you don't find anything at all. You don't find anything interesting. But if you look from, if you just go get little deeper, and if you look from a different angle altogether, then that leads to the solution. That leads to innovation also. This is for them is an innovation. They are able to solve the problem. Okay? Next. So, I have in fact several such examples. Almost all these examples I have experienced myself. So every day it is learning. Every day it's a new experience. Every day there is an opportunity to learn something new. There is an opportunity to apply your understanding, your knowledge in problem solving. So the key to success is observation. Observe carefully, then this observation should be coupled with thinking. Think critically and analytically. Then you should try to acquire a background knowledge of around that particular point. So you should read, you should go back to the literature and read the literature near about the problem. Next, every experience is a new experience. Never say no to opportunities. Don't ignore opportunities. Look for the opportunities. Next, do not hide the problems, highlight them. There is a tendency in everyone, I have seen almost in everyone, there is a tendency to hide the problem. If you hide the problem cannot be resolved. If you highlight it, then it becomes a problem and the problem can be resolved. Next, be with the truth. Imagination should be supported by evidence. So always one should be with the truth. And the truth will come out someday. Even if you try to hide out, it will come out someday. No prejudices or pretensions, only plain facts. This is very important to understand, that we should look at the things without any prejudiced mind. Otherwise, the new opportunities will not open up. Next, ultimately and finally, have patience. If you lack patience, nothing can be achieved. And my suggestion to the youngsters in particular, because I find the people lacking patience. Whenever I find a new person, he wants very quick returns. He wants to jump positions, he want to gain experience as much as possible in as small time as possible. That is not possible. If you have patience, then you live through the experience. You gain the experience automatically. And then you understand that then you understand the power of patience also. So, how to observe, how to make conclusions. For all this, one has to be patient. And patience, ultimately, patience means not doing anything is not patience. You should not concentrate on the result, the end result. You should concentrate only on the journey. The journey is beautiful rather than the destination. So keep this in mind and then go forward. You will definitely succeed. It will take time, but you will definitely succeed in life. This is my experience, and probably I should stop here. Thank you. Yeah, if there are any questions, I'll be definitely willing to answer. You can contact me later also, if required. Thank you. And more, very important, I sincerely thank the management, the principal, and the college staff for giving me this opportunity and presenting my thoughts. Thank you.
Cherish your words, sir. Observation is crucial tool in problem solving and fostering innovation. Thank you, sir, for adding immense value to our gathering today. Now, on behalf of everyone, we would once again like to thank our guest with token of memento for their valuable contribution and gracing us. I request all the dignitaries to participate in the felicitation program of Dr. S.S. Apte, sir.
Coordinator and Professor, Malla Reddy College of Pharmacy, to propose the vote of thanks.
Dr. Surya Devarkonda. With this, I would like to conclude first day's morning session with a vote of thanks. At the outset, on behalf of Malla Reddy College of Pharmacy, I thank Vinila Garu from Rakima Foundation. Very much honored, madam, for coming here. In fact, you have given us the opportunity to extend a little contribution in your service. We thank you so much. Sri Gurubhyo Namaha, Acharya Devo Bhava. Guru, Gu ante Gu means Gukaro Andakarasya Prakaro Tat Nivaranaha. Guru means darkness dispeller. పెద్దలు చెప్తారు, దైవ రుణం తీర్చుకోవచ్చు, పితృ రుణం తీర్చుకోవచ్చు, కొంచెం కష్టపడితే తల్లి రుణం కూడా తీర్చుకోవచ్చు, కానీ గురు రుణం తీర్చుకోలేంది వారి ఆశీస్సులతో ఎల్లప్పుడూ వృద్ధిలోకి రావడమే. అదొక్కటే మార్గం. డాక్టర్ డిఆర్ కృష్ణ సార్ అండ్ డాక్టర్ ఆప్టే సార్ వారిరువురు నా గురువులు. ఇన్ఫ్యాక్ట్ వారు వేల మందికి గురువులు, కానీ నా గురువులు అని చెప్పుకోవడంలో చిన్న పాజిటివ్నెస్. వారి యొక్క విజ్ఞత గురించి కానీ విశిష్టత గురించి కానీ చెప్పేంత జ్ఞానము శక్తి నాకు చాలా సాష్టాంగ నమస్కారం మాత్రం చేయగలుగుతాను. థ్యాంక్ యూ సో మచ్ సార్ ఫర్ యువర్ ఎన్లైట్నింగ్ talks today. I hope everybody have enjoyed. Dr. Tuljarani Madam Garu, Dr. Raj Kamal Garu, thank you so much. Dr. William Carey sir, thank you so much sir. I would like to extend heartfelt thanks to our beloved principal, Dr. Sudhakar sir. Thank you so much sir. Then I would like to, in fact, I'm very much, happy and lucky to be part of a very beautiful family here. Now my team members, I would like to extend my thanks to everybody. And thanks to all the dignitaries. Now it is lunch time, so please proceed for the lunch within 45 minutes. Next session will start. Thank you so much. Wish you all the very best. Vinila Madam Garu, wish you all the very best in all your future endeavors too. Thank you.
Thank you, sir. The next session of today's event will be held at Malla College of Pharmacy. Lastly, I urge all participants of poster, oral and scientific presentations from Malla College of Pharmacy to gather at the college to arrange for the same. Lunch will be served at MREC ground. The rest of the audience can join the lunch. Thank you all.